So thank you very much for being here in this, I think, wonderful session this morning. Please come up to the microphone if you have any questions, and you can also use the app where you can insert your questions, and then we will get your question here on the screen so we can also read it out loud if that's what you prefer. So I think we have a wonderful program this morning, and we start off with not the least of speakers, I would say, Seishi Ogawa. I think he hardly needs introduction. He has had a steady flow of seminal papers over the past 20 years, I would say. So it's very much looking forward to your presentation. Seishi, welcome.
Okay,
thank you for being here.
Good morning, everybody. And thank you to our chairpersons and for kind introduction. And also I'd like to thank all the organizers who are kindly inviting me to this symposium and giving me a chance to give a talk, which is something to do with molecular pathogenesis of MDS. This is my disclosures. So during the past decade, substantial advances have been made in the pathogenesis of molecular we are understanding of molecular pathogenesis MDS, which is primarily through the identification of the major driver mutations in MDS and other neoplasms. So this is one of the most updated pictures of the driver mutation in MDS, which shows these mutations are belongs to the several functional pathways, including RNA splicing, DNA methylation, chromosome modification, TP53-5Q pathways, and transcription factors, RAS, tyrosine kinase, and other signaling molecules, and cohesion complex. So this set of functional pathways are going to be have the primary role in shaping the development of MDS. And so these array of mutations, however, are not randomly acquired, but a set of mutations, particularly those involved in epigenetic regulation and RNA splicing, is going to be selected earlier stage of the MDS development and contributing to the development of coronary hematopoiesis, as you may know. And then the contrast, the other set of mutations are more likely to select in the later phase of coronary evolution, contributing to the progression to acute myeloid leukemia. So importantly, this body of knowledge about the gene mutation is successfully used to devise the novel molecular prognostic systems, which is now widely used for the clinical decision-making, contributing to the improvement of patient clinical outcomes. So this also used to establish the novel molecular taxonomy of MDS, which could be used to understand the underlying mechanism of the MDS, and also could be used for some stratification in clinical trials. So this is going to be presented tomorrow by Elsa Bernal. I just briefly explained this small subset of the MDS, which is characterized by the 17 derivative 17. So this is represents a very small subset of the IWG cohort, less than 0.5%, but this is really common in Asian population. You don't know, but this is. So 6%, Armenian 6% in Japanese population, in other Asian countries, but very rare in European countries. So you may not know this unique subset. So this is unbalanced translocation leading to the 3 copies of the chromosome 1q and 1 copy of the chromosome 7q. So this subgroup subset seems to have better prognosis compared to the -707q in other contexts. And so, in these cases, the more deaths from non-leukemic mortality, explained by non-leukemic mortality, particularly infections, compared to leukemic progression. So compared to the other subset, so this is very important to see the patient having this translocation. So this subset have the very unique co-mutation pattern. So more than 30 genes or chromosomes affected more than 10% of the cases, where the very high frequency with the transcription factor mutations are conspicuous, which is more than 6% of the cases, found in 6% of the more than 6% of the cases. So additional very unique feature is the high frequency of MIB1 and EDNK mutation, which is very unique to this subset, very rarely found in other subset, and likely associated with inferior prognosis. So finally, the TP3 mutation is very rare, or very, very rare, together with del5Q. This is highly contrast to the other del7Q in other contexts and other MDS cases. So this is probably explained by the MIB1 overexpression, as recently published from the Marie Sieber and her colleagues. So the MDM4 on the chromosome 1, 3 copies. So increase the expression of MDM4, which is the inhibitor of TP3. So they downregulate the PPT3 function. So actually, we observed the upregulation when we put in del517 and the downregulation of TP3. So this our MDM4 overexpression replaced the TP3 mutation function. So we also want to understand the mechanism of this translocation unique to the Asian population. So Dr. Yuichi Shiraishi, my long-time colleagues, decided to determine the complete sequence of the T2T sequence of this translocation. Because the translocation occurred within the highly repetitive alpha satellite in the both chromosomes, so he decided to achieve this using the long-read sequencing. So he found that the translocation occurred in the highly homologous alpha satellite in both chromosomes, in which complete match 20 base pair sequence triggered this translocation. He also developed a novel platform in which he can infer the structure of the centromere alpha only using the short-read sequencing. He applied this to the more than 2,500 of the centromere haploid from the 1,000 genomes project. He found that Asian population have a significantly longer alpha satellite than both chromosomes, the D1Z7 and the 7Z1, compared to the European population, suggesting that this large difference in alpha satellite lengths may at least partly explain this frequency ethnic difference in prevalence. Actually, the patient with the 1/7 translocation have significantly longer alpha satellite on both chromosomes compared to those without this translocation. So next, the probably driver mutation, the most intensively studied in hem malignancy, particularly AML and MDS, compared to the other solid cancers. But these driver mutations are less intensively studied using unbiased sequencing platform using, whole genome sequence. So we decided to analyze the unbiased detection of the whole genome, genetic alteration using the total of 913 patients with paired tumor normal DNAs using deep whole genome sequencing. So we obtained the 120x depth for the tumor and 30x for normal samples. So the mutation burden is 0.5 and 0.4 for AML and MDS respectively. This is comparable to the other solid cancer, many solid cancers, but significantly lower compared to the small cell lung cancer and other MS cancers and the melanoma. So, but the mutation burden is significantly differed between the TP3 mutation positive and negative cases. TP3 mutation positive cases, particularly those who have the direct TP3 alteration, have significantly higher burden of tumor mutation burdens, particularly structural variation is significantly more common in TP3 mutated cases. So we next look at the mutation signature to look at the etiology of disease mutations. So most mutations are explained by the clock-like signatures, and some SBS19 signature, which is whose etiology is still unknown. But this is not found in normal stem cells, so it's something to do with the hem malignancies. But what is complicated in this analysis is the small fraction of the mutation having these signatures, which is minority, minor signature, but it's all related to the chemotherapy. So conspicuously, 20 out of the 48 patients have the history of chemotherapy. So suggesting that when you look at the mutation signature, you can predict the history of chemotherapy. Because many people don't forget. So the history of chemotherapy is underreported. So this could be important in the future if you can do the whole genome sequence at a very low cost. So another things that deserve mentioning is SPS18 in AML cases. So this is uniquely associated with the core binding factor leukemia. So 821 translocation and invasion 16 both have these signatures. This is a hallmark of this translocation, which is something to do with reactive oxygen species. So we identified many novel driver mutations using the DNDCC analysis. These are positively selected in MDS, but also the frequency of these mutations very less than, many of them are less than 1% of the cases. So however, some non-driver mutation was successfully found using whole genome sequence, which is otherwise escaped the detection. Using the target capture sequencing. So many of them are related to the structural variations, which is, cannot be identified the target capture sequencing using in these many transcription factors and other cases. And also consequences focal amplification involving MYC and APO receptor. This is going to be exist as a ecDNA, so doubly minute, which is also escaped from the target capture sequence, and also deep intronic mutation involving TET2 and the M3A is also identified. So many of these mutations are incorporated in the IPSM system. So these patients, the grade of the risk might be changed using the whole genome sequence. Also, whole genome sequence can successfully detect the chrome strip cells. It is the so catastrophic event in which the chromosome is broken into the many fragments and rejoined at a single event. And then some of them escape from the rejoining and shuffling, and then amplified, selected as amplified double minute or ecDNA, and others are lost, contribute to the deletion of the chromosomes. So conspicuously, 70%, 5%, or 83% of the cases are related to TP3 mutations, but particularly biallelic TP3 mutations. So, but conversely, 30% or 40%, 30% of the TP3 biallelic mutation have these chrome slips. This is very common, particularly the EP3 APO receptors and ATP6 and ERG is a preferential target contributing the development of erythroid leukemia. So there, in summary, so using whole genome sequence, we can detect more abnormalities, which cannot be detected using target capture sequencing, truncating space, intronic mutation, focal opinion modulation, and blah, blah, blah. So finally, I would like to talk about the germline predisposition. This is emerging issue in myeloneoplasm because this is more frequent than previously expected. So using the whole genome sequence of paired tumor normal samples on more than 1,300 cases, so germline predisposition known pathogenic or likely pathogenic germline variant was found in 5% of the cases, of which by far the most frequent is DTX41. So this is explained 17% of all the germline predisposition, by far the most frequent. So the germline variant is very highly population-specific, different mutation, different population. By contrast, somatic mutations shared across the different populations. So the frequency, frequency mutation most frequent in high-risk MDS and secondary AML. So please note that 15% of the cases have only somatic mutation, no germline variant. This is need to ask some attention. So about 6% of the germline variant have the second hit somatic mutation, mostly R525HRL. So the penetrance was estimated using the king cohort analysis using fine-grained compensation. So the lifetime risk of the development hem malignancy is reached as high as 50%. This is not big, there is no big difference across the different mutations. So male is highly predominant and the risk is much higher in male. So this is only twice, but considering the only 10% are the female. So probably risk is about 10% larger in male cases. So combination pattern is also unique. CACSA mutation in DDX pattern is very conspicuous. And the difference between the before and after AML is not so conspicuous, suggesting no definitive event happening across the during progression. I'm sorry to poor figure. So this is also might be very important. IPVSS is not so effective to the risk of the risk prediction with these some positive cases. It can fail to discriminate the risk of the overall survival, so which is confirmed by the C index, significantly lowest index when this is calculated for the DDX41 positive cases. So instead, so there is a big difference in the clinical effect of truncating mutation with and without R525HR. So truncating mutation, patients who have the truncating mutation and R525HR have the most significant risk of the progression to acute myeloid leukemia and overall survival. So this is the future. So I was not able to explain the so some topics on inflammatory bone marrow environment and epigenetic abnormality, which is currently the most hot topics in this field. But I skip this. I'm sorry. So thank you for your attention. And this is my colleagues. Thank you for your attention. And I take any questions.
Thank you, Seishi, very much. So if there's questions, please come up to the microphone. Maybe I can start with one question. In the DDX41 families, there's, of course, the germline mutation, and then there's the second somatic mutation in a good number of individuals. But that second somatic mutation is present very often in a minor clone only. Do you have any hypothesis why this is not the event and is present in the whole leukemic clone?
I'm sorry, I have no good idea. But anyway, the presence of the second mutation significantly negatively affect the clinical outcomes. So probably the mechanism of the disease progression is very different from the DDX41 negative cases. Probably more epigenetic alterations or some bone marrow environment might be important to understand this pathogenetic, the molecular pathogenesis of this unique subtype.
Hi, Seishi.
Hi.
Hey, very inspiring talk. I was wondering, so in this proportion of patients where you see SBS 31, 35, so chemotherapy features, do you think that the chemotherapy-induced mutagenesis is producing any drivers or is participating in leukemiogenesis, or is it a passenger type?
It's a very important, interesting question. So unfortunately, this is so still we are analyzing in detail of these signatures with the association of the chemotherapy. I don't know, so I'm sorry, I have no good answer, so I'm sorry. So I really want to collaborate with you on this topic, because you're now embarking on the very big cohort to analyze germline predisposition. So cell-related MDS and ML.
So if there's no further questions, thank you very much, Seishi.
Thank you for your
and then we move on to Pedro Moura, who is from Karolinska Institute in Sweden, and he will talk about RNA splicing factor mutations and the effects on the hematopoietic proteome. So thank you very much.
Thank you very much for the kind introduction, and first of all, thank you very much to the organizing committee and to the MDS Foundation for inviting me to speak today. I just want to point out that I have no disclosures, and so I'm going to talk about splice factor mutations, and to do that, I would like to briefly introduce the background. Many of you know this, but there are several proteins which are critical, either from the spliceosome or as accessory splicing factors for the maintenance of RNA splicing. And in MDS, it is well known that as a group, these compose the biggest group of recurrent driver mutations found in equal or more than 50% of cases. They are also highly recurrent in other hematological malignancies as well as in solid tumors. So this is a highly prevalent and important group of mutations to understand. And when they occur, this has been well described by many authors in this audience and people who have organized this work, but they lead to dysfunction of RNA splicing, which I'll from now on call RNA missplicing. And perhaps the best well-known mechanisms for this are the dysregulation of SF3B1 mutations, inducing the generation of ring sideroblasts by missplicing of ABCB7 and other critical erythroid proteins for iron homeostasis, as well as the seminal work on SRSF2 from the Omar Abdelwahab group, showing that missplicing of one single transcript, the integrated subunit 3, leads to dysregulation of the entire complex. With that being said, the overall panorama of splicing events has been very well explored throughout the past decade, but we understand very little about the functional effects of these events. And in fact, even though SF3B1 we know the best of all, we know very little even in that context, and much less for the other ones. So this is springboarding back from previous work I presented in the previous MDS conference, where we showed that RNA and protein production were completely turned apart in the ring sideroblasts and during erythroid differentiation, but also in newer work where we investigated RNA missplicing in the hematopoietic stem cell, specifically using comprehensive single-cell RNA sequencing technologies, and we found extremely little difference in terms of RNA expression, but and you see here to the right, a very large amount of missplicing was happening between two SF3B1 mutations and against the general healthy background. So this leads us to one central question, which I'm trying to address in this work, which is, are the effects of RNA missplicing in the context of splice factor mutations primarily mediated by protein alterations rather than RNA alterations in itself? And so to do this, and from now on I will point out that this is all unpublished data, we started by designing a cohort to address this question. So we start from a broad cohort, primarily MDS patients, but including some secondary AML and CMML cases, who had cryopreserved mononuclear cells in large amounts, a complete clinical and genetic description, and a clinically and molecularly significant RNA splicing factor mutation. And this is a comprehensive cohort from Finland, from Sweden, and with the help of Seishi Ogawa from Japan as well. And you can see here the general cohort, 1/3 are SF3B1 mutant, 1/3 SRSF2 mutant, and the other third splitting between U2F1 and ZRSR2 mutant patients, as well as a healthy cohort for comparison. And so what we did was develop a method that enables the mini bulk analysis of FACS-purified cell subsets. And so to do this, here in the top left, I'm pointing out the analysis and purification background, but so we start from the broad mononuclear cell population. We then sorted CD34 positive cells as a broad hematopoietic stem and progenitor cell population, and then we purified specific compartments based on the prototypical cell surface marker expression. All of these were then subjected to mass spectrometry, totaling around 450 analyzed samples. This is the quality control data in terms of the depth and breadth of the proteome that we obtained, and you can see that the both of these were quite good. So we reached quantification of around 7,500 to 8,000 proteins in the broader populations. And in the subset populations where we can sort much less cells due to biological limitations, nonetheless, we achieved mean protein quantification of 7,000 or so proteins. And in more than 95% of samples, we have nearly 5,000 proteins quantified. So when we develop a method, we want to make sure that we agree with what it's showing. So here I'm showing the flow cytometry, representative flow cytometry data for one patient on how we do the sorting. And you can see here, I'm pointing out the 34 positive, 38 negative population as the multipotent progenitors, that then we go into the hematopoietic stem cell, which is CD90 positive, 45 array negative, and the MPPs, which are 90 negative, 45 array negative. And here is the digital sorting based on the mass spectrometry data that we have. And you can see in pink and purple the megakaryocyte erythroid progenitors and granulocyte monocyte progenitors on the top, which are 38 higher and 34 positive. And then the hematopoietic stem cells and multipotent progenitors together, which are slightly lower or negative in 38, as we can define it by mass spectrometry. Beyond this, focusing only here on the HSCs and MPPs, we then show specific and exclusive quantification of CD90 in all the HSC samples and in none of the MPP samples. So this is highly specific. At the same time, we want to assess the clinical relevance of this. And here I show just a summary of a few representative samples that we have with cytogenetic abnormalities in this cohort. And we can see here the protein level is quantified in the vertical axis across the length of the chromosome. In the protein that we can detect, we achieve the detection of the deletion 5q affected area as a significant downregulation of proteins specifically around the 5q arm. And in contrast, in monosomy 7 and trisomy 8, we see these changes across the entire chromosome in the direction that we expect. So the method is working in a way that is both biologically and clinically relevant as far as we were concerned. With that in mind, we first started by looking at the general clustering of our samples. And here, this is an abstraction because this clustering is based on the entire proteomic data, and here I'm showing only single sample GSCA analysis of the gene set signatures that we have here. So on the top you have a cell type enrichment analysis, and on the bottom the collection of misspliced genes so that we can see the general direction of enrichment of either higher expression or lower expression based on red or blue respectively. And you can see that if you look at the mononuclear cell fraction, this is essentially a very expensive way to do a comparative blood count or a bone marrow smear. But at the same time, and I want to point out the SF3B1 mutant samples in specific, which are red and sort of black here, that you see that if you look at the misspliced genes, you see an underexpression of these genes at the protein level, which already points out that you can find biologically significant signal even at the higher level of the hierarchy here. Going on a bit further, you now see that the clustering becomes much cleaner when we look at the general CD34 fraction, where now these samples cluster mostly by mutation, but also with some misclustering based on the cell type profile. And so if you go further, and this I'm not going to focus on in this talk, but then you can actually derive the signatures that enable us to understand what are the common and specific effects of these mutations in each of the differential cell subsets. So now I want to pivot to SF3B1 mutation specifically, which is the main topic of my talk. And here I'm showing the comparative protein abundance between SF3B1 mutant samples and normal samples. So I'm disregarding all the other splice factor mutations. And you can see that overall we have a large number of proteins that are significantly differentially expressed between the two conditions. If we now compare this against CD34 RNA data generated by Seishi's lab in another large study, we now find that there is extremely little concordance between the data. You can see in black the proteins that are significant in both RNA and protein. And in blue, the proteins are significant, that the genes are significantly differentially expressed at RNA level alone. And in red, the genes that are significantly differentially expressed at protein level alone. And you see that this is basically a cross. So that means that there is no concordance between these events. If we focus now on the misspliced genes specifically, we obtain a clearer picture of what is happening. So now, about 1/3 to 1/2 of the proteins are which transcripts are misspliced, are underexpressed in the SF3B1 mutant context. And I also think that it's particularly interesting that 2/3 are not. But if we then check this against the RNA data, we now see that even for those proteins which are significantly underexpressed because of missplicing, now we have a split in half. Half of those are cryptic missplicing events which induce nonsense-mediated decay, which is basically how most of us know the effects of SF3B1 mutations. But half of those do not. And in this case, that means that we have an invisible effect at RNA level. We can see the missplicing, but we cannot identify changes in expression. But now we can really find this as creating a knockdown of protein level, which is where it then becomes functionally relevant to the study of the molecular biology in the cell. Now, if we take the differential expression of these proteins as a whole, and here I'm taking all of the 500 or so differential proteins per subset, you can actually see that the organization of this is quite cell type specific. So in the left, you see the joint differential expression in the hematopoietic stem cells and multipotent progenitors together, and you see this high interconnectivity. And in large dots are the genes that are misspliced at transcript level, and in small, the ones that are not. And you can see that also the direction of expression, again blue or red, changes dramatically between the two types of subsets. In comparison, in the later megakaryocyte erythroid progenitors, which are further throughout differentiation, you see that the effects become more muted and in essence more crystallized. And this is where we find the ones that have been functionally characterized over the years. And now I want to focus just on two specific cases that we identified from our data just for SF3B1. But so this allows us to find complexes that are dysregulated as part of missplicing. And again, the large dots are misspliced transcripts. So here I point out the single-strand break repair complex comprised of these 4 genes. And you can see that RNA level, we know that 3 of them were already dysregulated at the expression level. But you find that one gene, XRCC1, is not. And in comparison, it becomes downregulated at protein level. This is relevant because XRCC1 is the bridge between PARP1 and the rest of the complex. And it is now well known from a study from 2023 that PARP1 inhibition is a sensitivity of SF3B1 mutations, but without a specific mechanism. And now we find a potential mechanistic explanation for this based simply on the underexpression of XRCC1 at the protein level. Another example is the WASH complex, where again we find one highly misspliced and highly differentially expressed gene, WASH-C5, at the RNA level, but few to no effect on the rest of the complex members at RNA level. And in comparison, all the complex members are downregulated at protein level. And there is quite an interesting study from several years ago studying only WASH knockout in the mouse, where the authors have shown that WASH knockout specifically induces an increase in competitive ability in the WASH knockout stem cells, enabling higher competitivity upon transplantation, as well as a conversion to long-term HSC fate, which in essence would provide a provocative explanation as to how SF3B1 mutations can peter out for a long time and become relevant in later age, which is another finding we have shown in the paper I mentioned before. And I want to mention also that this loss of the WASH complex that we see is one of very few events that are completely matching between SF3B1 mutations in the MDS context, in the AML context, and also not shown here in the CML context. So indeed we have a common molecular cause with potentially quite a severe outcome for the cell. So in conclusion, the low cell proteomic method we have established now enables the investigation of splice factor mutant effects beyond what had been achieved at RNA sequencing level, and we can do this even down to the hematopoietic stem cell, now moving beyond cell type bias and really understanding the molecular biology of the cell. We find that splice factor mutations in general, and here I show SF3B1, drive alterations downstream of RNA missplicing with ramifications that depend on this differentiated cell state. And by doing follow-up of the missplicing itself, we then find proteins that are changed without apparent change at RNA level, thus finding new mechanistic explanations for known phenotypes. So I want to finish by thanking my mentor, Eva Hellstrom-Lindberg, and the members of the group, as well as the members of my team, all our collaborators, but especially and our funding sources and the patients, but I especially want to thank the MDS Foundation. I was one of the fortunate 24 people who have been awarded the Young Investigator Award to pursue this research, and without that, this study would not have been possible. So thank you very much for listening, and I can take any questions.
Thank you very much, Pedro. Maybe I can also start here with a question. Is your mass spec methodology, does it enable you to also look at truncated proteins? So you're counting protein numbers, but it's possible, of course, that there are truncated proteins or otherwise altered proteins that would also give an extra layer.
Yes, so the data we have is at peptide level. So the protein quantification is a sort of average from all the peptide data that we have. So you can look individually at peptide level, and indeed we can find specific isoform differences, which is a much more complex analysis than I would present here, but also and relevant in the context of the 2/3 of missplicing events which do not change total protein level. You find the production of new misspliced protein sequences, which is highly relevant also given the cell study from the Abdel Wahab group 2 weeks ago on SRSF2. So indeed, this now enables that at unbiased high throughput level.
Right, so that's very interesting. I would have a second question maybe. It's long been known that these splice factor mutations are mutually exclusive in patients, and interestingly in this context, at the RNA level, the common targets that are suggested to be there were not identified. Are you now going to use the mass spec also to compare the different splice factor mutations to try to get to the pivotal targets that may be common to these splice factor mutations?
So that's a fantastic question. So the limitation we have currently is that all of our MDS or MN data is based on splice factor mutant MDS. So that means that if we would compare the common effects between them, we are not sure whether we are finding the common effects of splice factor mutations or the common effects of MDS, which then creates a complication. But that would be really interesting to identify what would be the common molecular targets. We have some inkling of what might be happening, but we would need to expand the cohort towards understanding that further.
Yeah. Michaela.
Yes, so thank you very much. Very interesting talk and a great talk. I have a question regarding SF3B1 mutation in MDS compared to AML. Previous work in your lab already shown differences related to RNA biology, and I wonder if at the level of the HSC or maybe CD34 positive compartment, you may find differences between sample you collect at MDS stage compared to AML stage.
So that's another great question for which we unfortunately do not have the data. So most of our patients, we have 22 SF3B1 patients. Only one of them, according to Elsa Bernard's IPSSM classification, only one of them is what we would call an SF3B1 high risk, sort of SF3B1 beta. So we don't have in this cohort SF3B1 AML, but that would be a fantastic question to ask from these samples.
Great talk. So I'm very much interested in the story about the XCCR1 missplicing related to the PAP sensitivity. So have you looked at the correlation between the alcohol consumption and SSVM mutation? Because alcohol metabolite to aldehydes, which induce double strand break. So probably I expect the heavy drinkers might escape from the development of the SSVM mutant MDS. So this is particularly interesting in Japanese population which have defective LD2 allo. So do you have any idea about that? Have assessed?
So how many of the SF3B1 mutant cases?
Because so the more alcohol consumption, the more aldehyde, the more double break. So this is harmful for the SSVM mutant cells. So the reduced incidence of the development of SSVM mutant MDS in the alcohol drinker, heavy drinker. So do you have any correlation between the alcohol drinking, alcohol consumption, and the SSVM mutant, frequency of SSVM mutant MDS?
So we've not looked specifically into that. So I unfortunately cannot really answer that, but I can add that what we see is also, and maybe this answers both questions at once related to XRCC1 and the potential for SF3B1 mutation have these different effects. We see very clear differences in terms of the surrounding proteome of this single-strand break complex and its effects in the HSC, specifically in the HSC, and everything else. So when we look at all the samples together, we see here the most, I'll just briefly cycle back. We see here the loss of the complex as a whole, but then related to that and to other also missplicing events, also described NSMC3A, which has been published before, we see a loss of the cohesin complex and of associated DNA repair pathways and cycling. Conversely, in the HSC population, we see the loss of this complex, but we see a very large increase in cycling in an abnormal way, which we cannot really trace to standard cell cycle progress. So indeed, maybe something very strange is happening in terms of cell division and what the cell is trying to push.
Okay, thank you.
Thank you very much. So if there's no further questions, thank you again for this wonderful presentation. And maybe
So it's my pleasure now to introduce the next speaker, Coleman Lindsley, who will discuss about the genetic pathways of disease progression. Please.
Good morning, and thank you for the invitation to come speak here. I want to start with an admission or a disclosure that is not financial, but rather personal, which is this title inspires a lot of frustration in me. And why is that? It's because I think I'm tired of being a spectator as I watch clinically patients continue to progress. And we have a scientifically valid and biologically beautiful sense of how gene mutations and pathways might collaborate to propagate disease and clonal advantage. But those scientifically valid observations, which I have dedicated a lot of time to studying, have limited, or I have to say, they have modestly realized their potential for patients. So good for papers, but not so good for patients quite yet. And so I think what I'll describe to you is kind of some thoughts driven by that frustration, but also driven by optimism that through our collaborative approaches and multinational interinstitutional approaches and industry academia collaborations, we might be able to realize that potential a little bit more. These are some disclosures that are not relevant to the discussion. So I'll start with two problems. One, this is just representative, not to pick on this drug in particular, but it's been a rough couple of years with pevonedistat, sabatolimab, magrolimab, tamibarotene, HMA backbone phase 3 trials that have been somewhat similar to this, which is negative. There's a couple solutions here to this problem. One is that maybe it's about trial design. Maybe we're picking the wrong patients or underpowering or looking at the wrong time. Maybe if we just included more patients, that little bubble there could have been had a p-value that was more inspiring. But I will propose to you that that's maybe not the solution in terms of trial design. The second problem is that I think we still have a limited understanding between the link between disease biology and treatment response in MDS, and even the difficulty in defining treatment response. So there's a lot of gaps there that underlie this frustration. And so expanding on that second problem, we know the parts list. We know what we're looking at largely. There are some new genes to be discovered. I know of some. And as Dr. Ogawa pointed out, there are pathways that are implicated: DNA methylation, chromatin modification, growth factor receptor signaling, DNA damage response, RNA splicing, cohesins. And these are not, as he said, combined in random order, in random combinations. And in fact, there is some order in that combinatorial diversity, and there is some order in those temporal dynamics with which they are combined. And I would say we can pick way stations where we have, at any given moment, a reductionist model for how these things work. And so maybe this is one. You can argue. I'm happy to argue with you about details here, but we can say there's initiation of a clone, progression, and transformation, 3 large buckets, and certain genes fall exclusively or mostly in one of these buckets. And by understanding at any given time what the gene mutations are and their relative abundance, we can map someone onto this progression. And as I said, it's kind of a beautiful process to be able to see that and measure that. But that beauty is not realized, I think, yet in terms of translation to patients. So how do we make it matter? What are our limitations? I'll describe one current approach that we're taking, which is essentially to really drive at how do we take an understanding of disease progression and make it relevant and translatable for patients. And so our efforts right now are largely focused on clinical trial-based analyses. So personally, I'm not really looking at single institution cohorts or ad hoc retrospective cohorts, except to generate hypotheses for which they're very, very useful for that. But clinical trials, in that they have prospective accrual, defined enrollment criteria, standardized treatments, uniform response adjudication, clinical follow-up, and the potential for appropriate sample collection are optimal sources of the evidentiary basis we need to develop refined heuristics that are useful for patients. And how do we study, how do we do this? I think academia industry partnerships need to be the basis of that, and that's through test development, conceptual frameworks, and therapeutic hypothesis testing. And then also what we're trying to do in the US is engage FDA with regards to assay standardization, defining contexts of use for results that we get from our analyses, and then how do we integrate these findings into regulatory pathways that will favor and propagate drug development and evaluation. So that's the mission here. And so one way that we're doing this is through the Foundation for NIH Biomarkers Consortium, which is a collaborative academia industry agency partnership led by myself, Chris Horgan, and Jerry Radich. And this is taking from the beginning to the end through development of standards and methods. But what I'm focusing on is clinical trial cohort analysis and regulatory engagement. And so one of the studies that we're looking at is the randomized phase 3 pevonedistat trial that I highlighted on as problem 1. And that was a negative trial, azacitidine versus aza plus pevonedistat. And the benefit here is that in collaboration with Amr Zaidan and Max Stahl and others, we have very refined and updated clinical adjudication of responses. And we also have about 1,200 samples from 400 and some patients enrolled on the trial. And this is just trying to depict to you the seriality of those samples and which ones they are. And so one is just the overall sample on the left, and on the right is kind of the samples per patient, kind of how we would look at mutations. And these are patients who went off trial early, and then those who stayed on longer have more samples. So the idea is baseline integrated with serial sampling to understand at any given moment if we can bin a patient or categorize them as more likely to progress or relapse or die, or more likely not to have those adverse outcomes. And so this is a picture you all would expect, which is this is the genetics of that cohort at baseline, lots of combinatorial diversity. These are the VAFs of here. And this was all done using ultrasensitive duplex sequencing, and so VAFs detectable down below 0.1%. And then, we can say, let's look at single gene associations with outcomes. It's also what you would expect, P53 at the top and DDX41 at the bottom in terms of high risk, low risk, and then a whole bunch of things in between. But I would say that single gene analyses with outcomes clearly, except for a couple rare cases, aren't really where things are. And so just to take an example of these, SRSF2 mutated MDS has itself many dyads and triads and tetrads that it of co-mutations. And this is just looking at for SRSF2. These are the significantly associated co-mutations. But then when we integrate that with outcome, it's not its most significantly co-mutated genes which are most important in defining which SRSF2 does better or worse. We use that clinical trial analysis, so we can say in the recursive partitioning analysis, it's actually the co-mutated B core, which is not significantly associated with SRSF2, but it is significantly associated with adverse outcome. And so when we look at here, SRSF2 in the overall cohort was favorable, and this is the HMA backbone, but when co-mutated with B core was unfavorable in that group. And so this is just saying, here are just the, if you look at RUNC, STAG2, ASXL1, it doesn't really discriminate subpopulations within SRSF2 mutated MDS, but B core and IDH2, for example, do. And so I'm not going to bore you with B core now, but let's just move on with more questions and answers. So the next theme will be all SRSF2 IDH2. And how do we move beyond the baseline where that dyad doesn't mean anything in terms of refined prediction of outcome, but it is clearly a highly recurrent progression mutation of SRSF2 mutated MDS. And so what I'll show you here is how do we integrate serial sampling to reveal different potential pathways or mechanisms of dynamic outcome assessment and prediction. And so one here is, this is SRSF2 ASXL1 IDH2 as the dominant clone. And then what we're showing you is serial sampling over the cycles of HMA-based treatment. And we can see clearly the PHS6 dominant subclone gets treated away, and a very minor 0.5%, 0.1% MIPL-P53 mutated subclone expands in its place. And so under the therapeutic constraint of HMA, a PHS6 subclone goes away and outgrows a transforming MIPL-P53 that was present at a very low level. So that's one thing that happens, subclone exchange driving progression. You can't really predict this from baseline. I'll tell you that a P53 at baseline does not correlate with P53 progression. You can take the similar patient, SRSF2 IDH2 RUNX1 this time, and nothing else on their duplex sequencing is totally clean below that. And then the moment you apply a therapeutic constraint of HMA, blossoms RAS-MAP kinase mutations. And we're not talking 1 or 2 or 3 or 4. There's many here after 2 cycles. And then all the 2 cycles, mutations are still present after 4, and there's more. And so this is a patient who, at baseline, I don't think we could have predicted this. And this is not the only outcome, but it is a recurrent outcome. But we don't understand the basic biology of why, in this patient, treating with HMA resulted in rapid outgrowth of these clones. And honestly, progression and death. So let's think again about context. And now we take another SRSF2 IDH2 patient over here. And let's say, how does that context inflect the interpretation or the outcome in an NPM1 mutant case? And so here we're just looking at, on this trial, NPM1 that occurs in a de novo context of DNMT3A, NPM1 RAS, and one that occurs in the context of a MDS backbone, the SRSF2 IDH2. And so here's NPM1. And so NPM1 goes away, very low level MRD that's detectable. I think that's like 8 cycles out. But in contrast, when the same therapeutic constraint is applied to the NPM1 in this context, nothing happens to NPM1 and outgrows a FLT3 CBP alpha mutated subclone. So there's consequence to context here in terms of how NPM1 in its progression, how its underlying context matters. So now let's say maybe the rules change when you change the therapeutic constraint. And this is going to start to get even more complicated quickly. So if we switch now SRSF2, IDH2 with some other mutations we know, but now instead of HMA, now let's just treat with an IDH inhibitor and see what happens. And in this case, we have a similar subclone exchange, and we can see this IDH2 RUNX1 subclone go away, and a CEPBP1 subclone grow out. And in this case, that was a beneficial exchange. And so, again, at baseline, a similar picture, but under a different therapeutic constraint, a different outcome. And so, a critical constraint that is very different as well for MDS in particular, because it's curative, is transplantation. And so, this is the Nakamura-Cutler led study, BMT CTN 1102, which is a donor, no donor analysis in older patients with high-risk MDS, where they showed that having a donor or actually getting transplanted was better than not having a donor or not getting transplanted. Similar to a study from Europe that showed, confirmed the same outcome. So now let's take IPSSM. And in this study, what we can see is just looking at the IPSSM groups, having a very high-risk IPSSM correlated with adverse outcomes independently for transplant in this cohort. So that seems well and good. But now let's look at when we go back and we sequence these patients, IPSSM very high is a heterogeneous group, biologically, genetically. And so in this cohort, it was roughly half driven by its P53 biology, and roughly half driven by a more typical splicing MDS picture that had normal karyotypes, relatively high blasts, and either KMT2A PTD or this SRSF2 IDH2 kind of RUNX1 picture. Why does this matter? It's because IPSSM very high is not valid for transplant as a predictive model using this in this donor, no donor analysis. And so here what you can see is this is validation of IPSSM for the non-transplant setting. So if you don't have a donor, rapid progression. This is concordant with how that model was developed. But when you, and in those, no donor, rapid death. But if you do have a donor, actually did very well. So in this setting, IPSSM very high, no P53, was predictive of transplant success. So low relapse or progression and good survival. Just highlighting that therapeutic constraints may modify how we understand patients and their risk models that were developed in different settings. And because PTD is a part of IPSSM and is clearly was an under-recognized progression pathway in MDS, we did some analyses in our local cohort. And so what we did is we, like I think most people are doing, we targeted chromosome 11, and using DNA-based sequencing, we're able to detect and quantify KMT2A PTDs, essentially by looking for increased representation of these tandemly duplicated exons within KMT2A. We also do split read analysis to quantify and identify the breakpoints for the tandem gain. And so this is a simple PTD where there's one copy gain of those 2 to 10 exons. It's not the only tandem gain, but that's one of the most common. But when we start looking in more detail, we can see that KMT2A PTD also has allelic progression that's associated with it. So there can be we identified a bunch of different ways in which the PTD itself became duplicated or progressed. And so here's via copy neutral LOH. Here's a gain of the PTD, now 3 copies. Here's a double gain, 5 copies. This is the relevant exons, and then higher order tandem gains, so many more copies. And this is consistent with the data that progression of KMT2A PTD is associated with higher RNA expression. And I think this is a relevant mechanism to explore mechanistically and to be able to track. And so when we look at these patients, we can also see that their copy ratio of PTD exons to non-PTD 11q23 goes up with progression from MDS to AML. And we can see in this is an exemplar case, we see acquisition of an allelic complex event augmenting the proportion of PTD exons correlating with transformation. And so just saying, yes, we know all the things, but maybe there are also things to learn about progression. And so hopefully this gives a general overview of kind of how at least one perspective on progression. And I really want to circle back and emphasize that to have actionable models about progression, I think we do need to accumulate numbers, thousands and thousands of patients treated on clinical trials where we have the ability to draw these clinical associations. And that does require partnerships with industry and advocacy from trial investigators and collaboration between the translational or clinical genomic folks and the clinical trialists to really advocate with drug companies for upfront inclusion of these things into trial design, but also into early phase clinical trials. And I have a lot of examples of that. This is obviously a team effort, and I'm happy to take any questions that you have.
So thank you very much, Coleman, for this very interesting presentation, which is now open for questions. So please go to the microphone. Perhaps I can start with one question. In our everyday clinical practice and also in clinical trials, basically we rely on variant allele frequency to derive clonal hierarchy and linear versus branching progression, which means that basically we do not take into account neither clonal hierarchy nor different ways of clonal progression to analyze the results of clinical trials and the molecular responses. So do you think that this might be relevant variables to take into account to improve our way to understand molecular response and clinical trials? And if in the affirmative, what approach do you suggest and you foresee to solve this issue?
I think biologically, it makes intuitive sense that in certain disease subtypes, it may be the founder clone biology that dictates outcome, and in other subtypes, it may be the progression clone that correlates with outcome, and that may relate to the disease or the treatment itself. I will admit that I lose traction in my mind when I start to try to think about all of that together, and my answer there is I want to let empiricism rule the day there and generate the data and develop models iteratively with these way stations of clinically relevant conclusions. And so I'd say, yes, I think clearly those, we can demonstrate those as being relevant. I think I have many more examples with IDH inhibition where the position in the clonal hierarchy seems to determine the nature and magnitude of response. And that is surely relevant in other places. But I do think we need to accumulate much more data and then source those data together through collaborative approaches, and then ideally use conforming platforms for analysis so that these can be translated and harmonized. So not a super concrete answer to your question, but I do think that's relevant.
So thank you very much, Colin. Very interesting. I think you're absolutely right. Treating MDS, we have to start to look after each package of treatment, each cycle, as you have done for AML for many years. Sorry. So what you show here is mainly higher risk MDS, where you have several mutations and they are exposed to sort of disease-reducing treatment. What I'm wondering is if you have thought of also introducing the cell stage, and this, sorry for my current cough, like for example in SF3B1 or in some other splice factor, or even in DEL5Q. I think there are several data, including from our group, showing that it's a matter of how many mutated versus wild-type stem cells you have. If you have enough wild-type stem cells, you will have, for example, a healthy erythropoiesis or erythropoiesis that can maintain itself. So are you also thinking about moving your VEF thinking, which I assume is mononuclear cells, down to the HSPC level to really understand where you have your mutations and how many? Because somehow, I mean, what we want in MDS is to have enough wild-type erythropoiesis, sort of hematopoiesis to maintain cell counts.
I think it's an important question, and I'll say that I'm trying to be as comprehensive as possible, but not trying to come up with comprehensive solutions, in part because I don't think we can boil the ocean, or any one person can boil the ocean, but I do think that if we can identify true reductionist models, then those can be the platforms to try to answer the types of questions that you're asking. So I think it's an important question, and then maybe we can answer those in focus settings like you're describing in SF3B1, and then use that to build out and nucleate working models that we can then translate across to a new context that's defined. And so I think the answer is yes, thinking about it, but no, not doing it now. But particularly if this is, again, a pitch for those of you who can influence clinical trial design, sample banking. You can't do that analysis if you don't have the right samples banked. And you can't link that to actual outcomes in a believable way unless those samples are from clinical trials, I believe. And so I think it's our duty to really advocate for that, to help achieve what you're describing, which is a definite goal.
Thanks.
Okay, so if there are no other questions, thank you again. And we now move to two selected presentations. So the first is by Dr. Onima Chowdhury, presenting a single-cell proteogenomic profiling of TP53 mutated myelodysplastic neoplasms. All clones are not equal. So, please.
Good morning, everyone. I hope you can hear me, even if you can't see me. So I'd like to start by thanking the organisers for selecting our abstract for oral presentation at this fantastic meeting. So I'd like to set the scene for the project, really, by telling you about a patient that presented to our team around about the time we were all really learning that there was a clear distinction between single-hit and multi-hit P53 with regards to patient outcome. She presented with MDS and a single P53 mutation and a normal karyotype with a low blast count and was classified as low risk. However, very rapidly, she developed an increasing blast percentage and a hugely complex karyotype, and sadly, she survived less than 8 months. So as a clinician looking after these patients, the obvious question is, can we detect this earlier? Is there a window of opportunity to capture patients like this one where we may be able to actually change the adverse outcomes? And that's really the fundamental question at the basis of this project. So we all know that following from wild-type hematopoiesis, typically a single-hit P53 mutation in a heterozygous state will develop, and then in the majority of patients over time, a multi-hit TP53 mutation will develop. And this will typically mean loss of the wild-type allele with either deletion of 17p, copy-neutral loss of heterozygosity of 17, or a second TP53 mutation. Now, there are two classification systems that we're all very familiar with that are broadly very similar, but there are distinctions. For example, with the ICC, a complex karyotype is part of the distinction for a biallelic P53 mutation, and it's a requirement that the VAFs are more than 10% for any P53 mutation for the ICC. And again, clinically, this can lead to discrepancies and problems when managing patients. But the core of the question we're really trying to answer today is, can early detection of early multi-hit clones enable and aid perhaps early intervention? So we have a single-hit population, but there are very likely to be undetected multi-hit clones within that population at a stage when we're classifying patients as single-hit. And perhaps at this stage, there would be a window of opportunity to change the outcome before a TP53 multi-hit clone begins to dominate. We also wanted to answer a more biological question, and that's really, how are these diseases evolving? What's the order of acquisition of genetic alterations? Is there a stepwise accumulation of events, for example, with a single hit, then a multi-hit, and then a complex karyotype developing in different cell populations over time? Or is there a more singular, catastrophic genetic event with all of these accumulating in the same cell population at the same time? And clearly, in terms of intervening, this makes a big difference. So in order to answer this question, we utilize the capability of the Tapestry single-cell DNA and protein sequencing pipeline. We designed a panel which covers not only TP53, but other myeloid mutations. But in the context of TP53, I think what's important is that we've carefully designed amplicons to be able to look for copy number alterations, particularly in chromosomes like 5 and 7, where we know there's clinically significant events happen. And we do that by designing amplicons across regions where we know there are heterozygous germline SNPs, which we can see are lost when there's an abnormal mutant copy number alteration. In addition to this, we have phenotypic information, so we can overlay our genotype onto the phenotype and also perhaps look to see if there are any early phenotypic differences in early multi-hit clones. So if we then move on to our bulk data, so far we have 21 patients and we've detected 26 TP53 mutations, as you can see here. Now, they're pretty typical mutations spanning the DNA binding domain of TP53. Looking in more detail at our cohort, but again, this is the bulk clinical diagnostics from the lab, we can split our patients into multi-hit and single-hit patients. Now, if you look at the multi-hit side on the right with the red, you can see that they're a pretty uniform bunch at this stage. But looking at the single-hit patients on the left, there are some discrepancies in the classification. And it's these patients where we're really going to focus our work, looking at these slightly aberrant classifications in a single-hit population. So moving on to our single-cell data, initially what we're able to detect is 10 distinct hematopoietic populations, which you can see here. On top of this, we can overlay our P53 genotyping data. Now, looking at this in more detail, if we compare wild-type to TP53 mutated cells, what you can clearly see is that there is an overrepresentation of P53 mutated MDS cells in the HSPCs and erythroid compartment. Now, what about when we split these into wild-type, single-hit, and multi-hit? So the wild-type cells are very much overrepresented in the lymphoid compartments. The multi-hit cells are overrepresented in the HSPCs and erythroid compartments. But interestingly, when you look at the single-hit patients, there is a much more mixed picture. Now, I'm now going to move on to talking about the genotyping data that we have collected. And I'll start with a few examples. So if you take this case 1, who has multi-hit P53 and a complex karyotype with a high VAF P53 mutation and WHO and ICC multi-hit. What do we see when we look at our genotyping data at the single cell level? We can see that it's very complementary to the information on the left. Here we have a small wild-type clone in blue and a very large singular dominant multi-hit P53 clone with a mutation and deletion of 17p, but we can also see that the monosomy 7 and the monosomy 5 are within the cell population, same cell population. Now, at the other end of the spectrum, we have a single hit patient who has a normal karyotype and a lower VAF TP53 mutation of 10%, but also an ASXL1 mutation of the same VAF. Now, if we look at our single-cell genotyping data, what you can see is that there is a large wild-type population, but there are actually two distinct clones, and the P53 clone is a single-hit P53 mutation, and there's a separate ASXL1 clone. Now, you'll probably notice that the VAFs are different or lower in the populations that we've detected with our single-cell sequencing, and that's probably likely due to the fact we're using frozen mononuclear cells, so you're looking at slightly different populations for genotyping. Now, our interest really lies in detecting early multi-hit clones. So is this technology powerful enough to do this? So if you take this third case, who's one of the 5 patients who did have discrepant single-hit classification that I showed you on the oncoprint. This patient has a TP53 mutation detected on bulk sequencing of 13%, but also a complex karyotype with a monosomy 7. So what do we see with our single-cell genotyping? And we can see a far more complicated picture than you might assume from that on the left. So a wild-type clone, but then it seems that there's sequential acquisition of single-hit TP53 mutations, then deletion 17p, and then monosomy 5 in one of the clones. So we definitely can detect early multi-hit clones in a patient like this. So I think so far, in terms of answering our questions, we can detect early clonal complexity with low-level multi-hit TP53 clones identified in patients classified as single-hit disease. In terms of the biology of the disease and the order of acquisition of genetic alterations in our cohort so far, we can see that copy number alterations typically occur simultaneously within the same clone or after the acquisition of a second TP53 hit. In terms of moving this forward, our ultimate goal really is to develop a useful early detection tool. So our next steps, as we've talked about with some of the other talks, is definitely to expand the cohort, focusing on single-hit patients and looking longitudinally, ideally in the context of clinical trials, and really figuring out which patients could benefit from this early detection of multi-hit clones. For example, those with predisposition syndrome spring to mind. And I think this meeting is a really great opportunity to talk outside of these talks now, and it'd be great to hear everyone's thoughts on how they think we could move this forward and to build some good collaborations. So thank you very much for your time, and I'd particularly like to thank Chris and Amy, who have taken on leading this work while I've been seeing the patients. Thank you very much.
So thank you very much for this very interesting study, which is open for discussion. So please, first.
Yeah, thank you, Oni, for the great talk, beautiful data. Just 2 questions. So you see the distribution of TP53 mutant clones, say, more restricted in HSPC population in double hits, but single hit is a mixture type. And in wild type, the cells are more dominant in lymphoid populations. So do you think there's a dosage effect on this distribution?
I see what you mean. It's possible. It's possible. I think, to be honest, I mean, our single hit, we have far fewer single hit cells than we do multi-hit cells. And we can see that it's not biased just by a single patient. That's the other thing to think about when you present UMAPs like this. So it's possible there's a dosage effect, but I think the cohort's still pretty small, so I think drawing too many conclusions from that would be a bit tricky, but that is certainly one of the things that we're thinking about.
Yeah, thank you. And the second question would be, say in the MPN context, Adam has previously shown that it was the inflammatory environment that drives the expansion of TP53 mutant clones.
Yeah.
What's your expectation to see in the MDS context?
I think there will be differences in the different myeloid neoplasms. As you say from Alba's paper, the erythral bias and the skewing that they saw seemed very much inflammation-driven, which makes sense in MPN. Definitely possible in lower-risk MDS. I think there may be some similarities. But also in MPN, the acquisition of events is quite different. Often they start off with a different, a driver mutation, then develop P53. You don't tend to get these sort of catastrophic karyotypic changes quite in the same order. So I think it would be really nice to compare these different cohorts as well. Thank you.
Okay, so we have time for 2 very quick questions and quick answers because we are running a bit out of time. Please.
Perfect. Very nice talk, David, from Moffitt. One quick question. So out of your, I think, 21 patients, how many didn't have any multi-hit? Was it only 2 that I saw from the abstract? Just curious the percentage of patients that truly have no multi-hit at, let's say, at the single cell level. Could you comment on that?
So who didn't have multi-hit at the single cell level, as I said, we had 5 that were discrepant. Everyone else had multi-hit clones. If the bulk sequencing found multi-hit, we found multi-hit. In the 5 discrepants, 3 very clearly have multi-hit clones. The 4th, it's sort of at that sort of QC level where we need to look at it and probably run it again. But 3 to 4 out of the 5 have got early multi-hit clones for sure.
And yeah, I think potentially just sometimes maybe we're overemphasizing where a majority of patients are the very high risk. I guess just similarly, and probably the numbers are too small, it'd be nice as your cohort expands. As I don't know if you had a couple of cases, based with a different blast, let's say as blasts do get greater than 5. I'm curious, the couple cases that they were all truly low blast, I would assume too, less than 5%?
Yes.
Thank you.
Okay.
Thank you.
Very quick question. Thanks for your presentation. On those discrepant cases that had small multi-hit clones, did you have any sequential samples? Because we expect those to really expand or follow up,
Absolutely. So I think looking longitudinally is going to be absolutely key. And again, looking in the context of clinical trials would be great. So we don't on those specific patients, but in terms of building our cohort, that's exactly what we want to do. Thank you very much.
Thank you very much. And then we move on to the last speaker, who is Stephani Schmitz, who is working actually here in Rotterdam. So welcome to the stage. And she's going to talk about metabolic reprogramming in del(5q) MDS.
Well, thank you very much for having me and for selecting my abstract for an oral presentation today. So I'm going to talk about del(5q) MDS, specifically with casein kinase mutations today. I have no disclosures. So del(5q) MDS is known as one of the most common cytogenetic abnormalities in MDS, and what we have there is the case of deletion of one long arm of chromosome 5 that leads to a haploinsufficiency of a set of genes, and this loss then drives a very distinct clinical phenotype and also a clonal expansion of the mutated cells. But about a decade ago, our group, alongside others, have first described recurrent mutations in the remaining copy of casein kinase 1A1, also describing them as the first recurrent mutation in del(5q) on the 5q- arm. And these mutations occur in roughly 6 to 18% of del(5q) MDS cases. And although the cohort studies so far have been very small, they've been linked to lenalidomide therapy resistance and also adverse disease outcomes. Zooming into the mutation distribution on the casein kinase gene, it's very apparent that there is a clear hotspot in the E98 amino acid, which sparked our interest in looking into this further. So we studied this mainly in the mouse so far. Of course, we plan to expand it in a human context or a cohort, but coming across these patient samples is a little harder. So in the mouse, what we did is make use of a conditional casein kinase knockout in hematopoietic stem cells, in hematopoietic cells specifically, and then we transduced these donor cells with a retroviral plasmids encoding either wild-type or E98V version of casein kinase, which is an example of one of these hotspot mutations. Then we transplant them into recipient animals and excise the endogenous copy to only look at the effect of the mutation. And what we see is when we just look at the mice with an empty vector, so casein kinase 1A1 knockout hematopoiesis, they have bone marrow failure, which has been described previously, but with the mutation, this can really be rescued, pointing out that the E98V mutation is not a loss of function mutation, but rather an altered function. Then we were interested in looking into whether this has an impact on the HSC function, especially in the context of casein kinase, which has been described previously to be a main driver of the clonal expansion at haploinsufficient levels. So we performed a serial transplant using the same mouse model that I've described before. And what we see is that there's not really a difference with the mutation comparing it to the wild type. So yeah, looking like the E98V mutation does not add to the clonal expansion that the haploinsufficiency has. Even though we didn't see an impact on this proliferation clonal expansion part, we did zoom into the HSC compartment and see that when looking at the LSKs, we have a significant expansion of CD48 positive, CD150 negative HSPCs, pointing into a direction that there is not a self-renewal phenotype, but rather a differentiation phenotype with increased occurrence of more lineage-biased progenitors. We then performed bulk RNA sequencing on these LSKs from the mouse transplant, and in this principal component analysis on the left, you can see that they have really distinct transcriptional profiles. And then we zoomed into this LSK compartment more specifically by performing a cybersort deconvolution analysis to look at the composition of the HSC compartment. And what we can see is that there is slight differences in the long-term, short-term HSCs, but if we move further down the hematopoietic hierarchy towards more lineage-biased progenitors, you start seeing more apparent effects with the mutation. Specifically, reduced megakaryocyte progenitors and an increase in lymphoid progenitors, really highlighting that we, with the mutation, seem to have a HSC differentiation bias. To then have a more controlled platform and look at the specific mechanisms that are at play here, we generated an in vitro model system using immortalized murine hematopoietic stem and progenitor cells with either the wild type or the E98V copy, and then performed coupled RNA sequencing, proteomics, phosphoproteomics, and kinase activity profiling to get an in-depth picture of the molecular underpinnings that the mutations have. And starting by looking at the kinome activity profiling, we see that with the mutation, here comparing it to the wild type, we have a clear effect, downregulation of casein kinase 1A1 activity specifically, but there's also a really broad suppression of other kinase families as well, really hinting towards the mutation having a really broad effect on signaling cascades. One thing that was really noticeable as well was a very prominent downregulation of ribosomal gene expression, as you can see in the heat map on the right. And then functionally, we linked this to decreased protein translation rates by looking at OPP incorporation, which was significantly lower in cells with the E98V mutation. And we thought it was particularly interesting thinking about the del5Q MDS phenotype, where I here highlight RPS14. RPS14 is also found on the deleted arm and has been shown at haploinsufficient levels to really drive the disease phenotype as well. And in this context, the 5q minus syndromes have also been described as a ribosomopathy. And here we can show that the E98V mutation seems to really reinforce this phenotype that we see, and might compound on this effect. Seeing the decreased or deregulated signaling networks and also the impact on the ribosomal gene expression and translation rates, we were prompted to look into metabolism. And what we did here is perform a principal component analysis on our data, both on the transcriptomic and the proteomic level, looking or clustering the samples based on either glycolysis or oxidative phosphorylation genes. And you can see that they cluster very distinctly, really pointing towards very drastic changes when it comes to metabolic gene expression signatures. We then looked into this on a more functional level by performing seahorse analysis, and what we found was that cells with the mutation have an increased proton efflux rate, suggesting that they rely more on glycolysis, and in line with this, the mutated cells have a decreased oxygen consumption rate, showing that there is a metabolic switch from more mitochondrial oxidative phosphorylation towards glycolysis. We then were prompted to look into reactive oxygen species and measured cytosolic as well as mitochondrial ROS, and could show that these are significantly lower, which is in line with less oxidative phosphorylation, which generates a lot of ROS as a byproduct. This fit nicely together. And then we were interested in looking into iron metabolism, with this being really a central player with looking at mitochondrial function, but also ROS generation. And what we found was that at baseline, the cells with the mutation have increased levels of the iron importer and exporter, really highlighting that there might be a dysregulation of iron handling with these mutations, which is specifically interesting thinking about del(5q) MDS as well, as these patients have anemias and often get blood transfusions, which then lead to, in severe cases, to systemic iron overload. To mimic this in vitro, we treated the cells with the mutation and the wild type with iron, with exogenous iron, and could show that they are very susceptible to this, with decrease in viability and an increase in reactive oxygen species. Thinking about this and seeing this effect in response to iron, we were prompted to look at ferroptosis, which is a non-apoptotic iron-dependent form of cell death that is characterized by uncontrolled lipid peroxidation. So what we did is treat the cells with Avastin, that's a compound that induces ferroptosis. And what we found was that we have a very profound downregulation of ferroportin, the iron exporter, at the same time as an increase in cytosolic ROS levels, but most importantly in lipid ROS levels, really all highlighting that these cells are very specifically sensitive to ferroptotic cell death. And this already brings me to my summary. So what we could show in our studies was that the casein kinase mutation seems to rewire HSC biology in a very nuanced way. On the one hand, we seem to have sort of a stress adapted response where cells have downregulated ribosomal gene expression and lower translational rates, a shift from glycolysis, from oxidative phosphorylation to glycolysis and lower ROS levels, which is more in line with a more protected HSC state, but on the other hand, we see that the cells, rather than relying on self-renewal, they seem to have a differentiation bias, specifically in the megakaryocytic and lymphoid trajectory. Really highlighting that the mutation leads from a metabolically preserved state to also a biologically differentiated state. But specifically, what this leads to is an iron imbalance that can be therapeutically harnessed by having this altered iron metabolic state that renders these cells susceptible to ferroptotic cell death, which could provide a novel therapeutic intervention in the case of del(5q) MDS. And with this, I want to thank my lab, specifically Rebecca Schneider and Elaine, for supervising me as their PhD student. But I also want to highlight Juliette Pierce, who's a postdoc bioinformatician who has worked very closely together with me on this project. And thank you.
Thank you very much. Are there questions from the audience? One coming up. Go ahead.
Thank you for a wonderful presentation. I'm just wondering, based on your fantastic data on iron imbalance, if these patients with this mutation compared with the del(5q) without the mutation have a higher percentage of ring sideroblast production, given that this is a phenotype in sort of double TP53 and del(5q. Do you see any evidence for this being the mechanism for ring sideroblast production in such patients?
That's a very good question. I don't have a very clear answer because we don't have a lot of cohort data looking at this. I know I have spent some time at the MLL in Munich where I had the opportunity to look at their cohort where they also profile casein kinase mutations, and there was an increased association with SF3B1 and ASXL1 mutations, so maybe this could correlate to this, but we just don't have enough data. We're working on collecting more clinical data. Not everybody profiles casein kinase mutations in their myeloid panels, which also makes it harder, but hopefully we can answer these questions better in the future.
So we're running into the coffee break, but there's room for one quick question. And a
very interesting talk. So I just wanted to know the effect of the mutant casein kinase on the Wnt pathway activation. So it is included in the destruction complex, and I'm very interested in its effect on the beta-catenin application of the beta-catenin on the Wnt pathway signaling. What is the effect on Wnt signaling, up or down, or no effect?
Okay, yeah. We have shown previously that the mutation, so the haploinsufficiency leads to very clear upregulation of Wnt signaling, but with the mutation we also see that. So higher beta-catenin levels in the nucleus and upregulation of Wnt signaling, which could also contribute Wnt signaling and beta-catenin specifically has also been shown to impact the switch from oxidative phosphorylation to glycolysis. So I think everything we see is probably driven by upregulation of Wnt signaling.
Thank you.
Okay, thank you very much. With this, we end this session, and we welcome you back at 10:50, I think, after the coffee break. Thank you very much for all the speakers.